experiment is about water and humus. So we're going to see how we can carry out an experiment to confirm that actually soil contains water. To show that soil contains water and humus. We put them together because the procedure is quite the same. We can obtain both of them with the same procedure. So what shall we need? And those are the materials and apparatus. The soil itself. The procedure, how shall we carry it out? So we pour about 5 cubic centimeters of soil into the crucible and measure the two. So we measure and record the weight of the two, that is the soil and the crucible. Next, we then heat the soil with a Bunsen burner. So the use of the wire gauze is to support the crucible on the tripod stand. And the tripod stand supports the setup above a Bunsen burner as it heats. So we light the Bunsen burner and start heating the soil. Now at intervals of time, we remove the soil and measure and keep measuring the weight of the crucible and the soil until a constant weight is reached. So as you continue heating and weighing, there reaches a time when there is no more decrease in mass. Actually, the mass keeps on decreasing. And why is it decreasing? Because as we heat, the soil, the water that's found within the soil evaporates off, and that causes a difference, a decrease in the amount of soil. So, of course, the amount of crucible doesn't change, so it's only the amount of soil that changes due to the loss of water from the soil. Now, a new weight is reached, and that weight is because all the water has been lost. Now, let's assume that we had a weight of soil which is X grams, and the new weight after heating and there's no more decrease happening, let's give it Y. We can find the weight, the mass of water that was lost by evaporation by finding the difference. And we can do that. So how do you find the amount of water, the mass? Remember, the mass of the crucible never changes. It's the mass of soil that reduces because it has lost the water that it contained initially. Therefore, we gave our initial value X grams. So this is going to be X grams minus Y grams. In that case, we are able to find the amount of water. Let's give them numbers. For example, if the mass initially was 17 grams, let's assume that X is 17 grams and Y is 14 grams. Then the mass of water will be X minus Y, which is 17 grams minus 14 grams to give us 3 grams. That means that water constitutes 3 grams of the total weight of soil. That's about water. Our next experiment is to show that soil actually contains living organisms. Most of them we cannot see with our own eyes, but you can take it for a scientific fact that they actually are always there in any amount of soil, in any type of soil. So what do? We measure about 5 cubic centimeters of soil, tie it in a muslin bag. A muslin bag is an equipment we use in the laboratory. It has small pores through which small gases can pass through. And so we pour it in a muslin bag and tie it with a thread. After tying it with a thread, we suspend it over lime water, and we close the whole content with a cork and cotton wool that covers the hole within the cork. How does that look like? We're going to see. So we tie it as I'm going to draw so that we can see it, and we'll let it suspend for some time. We actually do the same with another soil sample, but before that we first heat the soil sample with over a Bunsen burner to make sure that if there are any organisms there, we kill all of them, and then we repeat the same procedure for that sample of soil. So we repeat the same procedure, but before that we first heat the second soil sample, and the diagram will look like this. These are our two samples. We have used test tubes in this case. So this is the cork. There are types of corks. There are those that have holes through which maybe tubes can go through, and if such a cork is what one has used, we cover it with cotton wool so that there is no space whatsoever where the air can pass. But if there are those that do not have a hole in them, those are fine. You don't need any cotton wool with them. This is our thread. This is our muslin bag. This is our test tube. And this is our lime water. The only difference with these two samples is that one of them was heated before and the other wasn't. So this was, let's call this the heated soil or baked soil. And let's call this the fresh soil. So we leave this experiment for some time while observing. Now, after some time, we shall see that the lime water here just remained colorless. It's a clear liquid, it's more like water. But we shall see that the lime water in fresh soil turned milky. Why did it turn milky? Because lime water in science is used to confirm presence of carbon dioxide. Now, the living organisms within the soil that was not heated keep carrying out respiration. Now, respiration uses up oxygen and gives out carbon dioxide. The carbon dioxide goes through the muslin bag to the lime water, making it to turn milky. How about in this case, we do not have organisms were killed due to the heating of the soil. Therefore, no more respiration is taking place. Therefore, no more carbon dioxide is produced, and for that reason, lime water will still remain clear. So the observation here is lime water remains clear, and here lime water turns milky. Why do we put a cork above the test tubes? To prevent any gases like carbon dioxide from entering to cause change of color in lime water. That's why even if it has a hole, we tighten it with cotton wool. And that proves that soil contains living organisms because they're the ones that keep on carrying out respiration.